Hello, this is Dr. A with a review video for clinical chemistry on sodium, chloride, and potassium. Alright, so let's start with sodium. Sodium constitutes the major cation in plasma and extracellular fluid. You get sodium from salt. And as a side note, do you know that the biggest source of sodium in the American diet are breads? That's interesting, in doughs and stuff like that. Active transport systems such as your sodium potassium pumps, they prevent sodium equilibrium in all cells. It would be equilibrium would be the same amount of sodium inside and outside of the cells. That's not how sodium is distributed. So these pumps actually keep pumping all the sodium out of the cell and put it in plasma and extracellular fluid and putting all the potassium inside the cells. The regulation of sodium is maintained by the kidneys, and the main hormone is going to be aldosterone. Now, aldosterone is produced by the adrenal glands, and then it goes and affects the kidneys, and aldosterone triggers the sodium reabsorption in exchange for potassium. So what aldosterone does is take sodium from the urine and puts it back into the blood. And then it takes potassium from the blood in exchange and dumps it into the urine. Okay, and so, that's what it does. When it moves sodium, then water follows and chloride follows. So it's a way to put more volume back into circulation. And that's when aldosterone usually kicks in, is when you see dehydration. The function of sodium is to maintain the osmotic pressure of extracellular fluids. So again, your interstitial fluids, joint fluids, and stuff like that, but also plasma. And it is, another role is to maintain acid-base balance and blood viscosity. So it has roles in that. Transmitting nerve impulses. So with the sodium potassium pumps and channels and stuff, those are very important in nerve conduction, and they also assist in muscle contraction. So very, very important electrolytes. It has a narrow reference range, and slight changes can have significant impact on patient's health. The reference range for sodium is 136 to 145 millimoles per liter, or milliequivalents per liter, depending on how you measure it. So in clinical significance, hypernatremia or high sodium levels are less common, but they're seen in severe dehydration, especially if you have excessive sweating where you're losing salt, right? Hyperaldosteronism, so making too much aldosterone, so that would make sense because it would put sodium back into the blood. And then Cushing's syndrome, where you have excessive amounts of sodium that are reabsorbed also because you have excess excretion of cortisol and all that from the adrenal gland. And the other one is hyponatremia. So hyponatremia is a low level of sodium in the blood, and this is seen in cases of decreased intake, vomiting and diarrhea. And then it can be delusional in liver or heart failure when you have a lot of volume of fluid that is accumulating in different places. And of course, also can be seen in ketoacidosis. The lab procedures for sodium, so generally it's measured by ion selective electrodes. That's the most common, also known as ISC. So the principle of ISC is that there's a semi-permeable membrane and two electrodes, a reference electrode and a sample electrode, and a potential is produced due to the different ion concentration on either side of the membrane, so on the reference and the sample, and that difference in potential between the two can be measured and then correlated with the concentration of ions in the sample. And what changes is the semi-permeable membrane, and that determines what ion you measure. In direct ion selective electrodes, you use an undiluted sample. Those are often seen in point-of-care instruments and ABG analyzers. And then the indirect ISC uses a diluted sample, and that is the one that's run in most large analyzers. So the sources of error for sodium, because the indirect IC uses a diluted sample and it's prone to interferences by both high lipids and high protein concentrations, and these substances can displace serum or plasma and yield falsely decreased results. And the values for sodium. The electrodes can be coated by a protein from the sample, and also source of errors, then usually it's, if they can't be cleaned, usually they have to just simply be replaced. The specimen types for sodium are whole blood, serum, and plasma, usually lithium, heparin, and urine. So chloride is sodium's buddy. It is the anion of highest extracellular concentration. So again, because that's also where sodium is, that is where chloride is. So extracellular means interstitial fluid and also plasma. One of its main jobs is to maintain electrical neutrality. And so it moves passively with sodium. So it's sodium's buddy, follows it. So where sodium goes, chloride goes. And it tends to move against bicarb to maintain electrical neutrality in the red cells. I'm going to explain that one a little bit more. And of course, the functions are then osmotic pressure maintenance and electrical neutrality maintenance. Usually the same conditions that lead to hypernatremia can lead to hyperchloremia, and the conditions that cause hyponatremia will cause hypochloremia. So let's talk about the chloride shift. So this is related to maintaining that electrical neutrality. It is related to the transport of CO2. So in the lungs, chloride is going to move to the plasma, and bicarb will move into the red cell. So chloride and bicarb swap places. So bicarb goes back into the red cell where it picks up the hydrogen ion that's hanging out over there with hemoglobin, and combines with hydrogen ion, it becomes CO2 and water, and then CO2 can cross the membrane of the red cell and be exhaled into the lungs. At the tissue level where CO2 is generated, the red cells that pick up the CO2 then will dissociate it. They'll keep the hydrogen ion and tag it onto the globin part of hemoglobin, and they will kick the bicarb ion out of the red cell into the plasma. But then, of course, when they move that out, they have to replace it with chloride from the plasma. So they do a switcheroo every time when they arrive at the lungs and at the tissue level. So this diagram kind of explains it. It shows at the tissue level, but the opposite happens at the lungs. So tissues are generating CO2. CO2 combines, enters the red cell, combines with water, generates carbonic acid with the help of carbonic anhydrase, and makes bicarb and hydrogen ion. The hydrogen ion again binds on the protein portion of hemoglobin. Bicarb is kicked out and hangs out in plasma, while chloride is brought in exchange for that bicarb. And then they float, and then you can do the opposite for what happens at the lungs. So the lab procedures for chlorides, your specimen types are serum, plasma, and whole blood, and also urine. Although it's less commonly measured on urine, it can be done. We can also do spinal fluid. So chloride in spinal fluid has a higher concentration than in serum, and is due to the lower protein concentration in spinal fluid. In bacterial meningitis, there's a higher protein concentration in spinal fluid, and thus the chloride concentration will be lower than the reference range for CSF. It is, however, not commonly measured in spinal fluid. And then the other one is sweat chloride, but basically that was the old test for cystic fibrosis. It's been replaced by genetic testing, but there's still some labs that can perform sweat chloride test, and patients with cystic fibrosis have higher levels of chloride in their sweat. So the lab procedure is again an ion-selective electrode with a membrane that is selective for the chloride ions. The principle again is the same. The ISD has a semi-permeable membrane, 2 electrodes reference and sample. The potential is produced due to the difference in the ion concentrations on either side of that membrane. The difference in potential between the two can be measured and correlated with the concentration in the sample. And so that's exactly the same as for sodium, and it can also be direct and indirect. The reference range is 98 to 107 millimoles per liter, or milliequivalents per liter if you use that. The sources of error, so a potential source of error is chloride from tap water or on glassware used for chloride testing. So that has to do with the quality of water and if your analyzer uses water and stuff. So the next one is potassium. Potassium is the most abundant intracellular cation. So potassium lives inside your cells, most of it. And so again, 98% of the potassium in your body is inside of each and every single one of your cells. Your sources of potassium are fruits and veggies. Bananas are a really good source of potassium, but you can find it in fruits and veggies. If you don't like bananas, don't worry, just eat fruits and veggies. The function of potassium is the regulation of neuromuscular excitability, so having to do with impulse conduction in muscular contractions and stuff like that. The contraction of the heart, the interstitial fluid volume, and hydrogen ion concentration are all influenced by potassium. Potassium has a role in all of these. And as a matter of fact, low levels of potassium can give you like muscle cramps and stuff like that. Aldosterone is involved in the kidney's regulation of potassium. So as I explained earlier already, since aldosterone is released usually in times of dehydration, and the idea is that it moves sodium from the urine back into the blood. Water follows from the urine back into the blood, so increases blood volume. But in exchange for pulling that sodium, it has to kick potassium out. So high aldosterone will drop your potassium levels in your blood. Factors that will influence the distribution of potassium. So any inhibition of that sodium-potassium pump by certain conditions will affect obviously the concentration of potassium. Insulin promotes potassium ions entering muscle and liver cells, right? Your catecholamines, which are like epinephrine, norepinephrine and stuff, they promote the cellular entry of potassium, so move potassium where it belongs. Exercises will increase plasma potassium. So remember, potassium is not really supposed to be that much in plasma, right? So it moves it from the cells into the plasma. Hyperosmolality will deplete potassium, and cellular breakdown releases potassium into the extracellular fluid, which then it can, be cleared by the kidneys too. So hyperkalemia, or a high level of blood potassium. So one of the consequences of hyperkalemia is it can paralyze the heart muscle, cause bradycardia, so very slow heart rate, mental confusion, and numbness. On a side note, lethal injections are an overdose of potassium. Hypokalemia in homeostasis will occur when potassium leaves the cells faster than it can be excreted by the kidneys. It is seen in conditions such as dehydration, shock, metabolic acidosis, acute renal failure, chronic kidney disease, and rhabdomyolysis. Artifactual hyperkalemia, that means it's basically not really caused by a disease process. It occurs in patients with high platelet counts. Platelets will release potassium during the clotting process, and then it also is evident in hemolyzed specimens because potassium is released from the cells when those cells are hemolyzed. And then it's artifactual if the hemolysis happened in the tube and is not happening actually in the patients. And then hypokalemia, which is a low potassium level, the consequences of hypokalemia are an irregular heartbeat and tachycardia. Fast heartbeats are the opposite of hyperkalemia, right? And it can lead to cardiac arrest and muscular weakness. So high potassium and low potassium can both be really dangerous to a patient's health and can be lethal. What can cause hypokalemia are decreased intake of potassium, so that would be through decreased intakes of fruits and vegetables and stuff like that, or an increased loss through the kidneys, especially if there's high aldosterone, but also an increased loss by, for example, through vomiting, diarrhea, any kind of GI issues, and of course a diuretic use, which promote the loss of water and stuff in urine, and then potassium will be lost with it too. It is also seen in Cushing's syndrome, hyperaldosteronism, metabolic alkalosis, and salicylate use. The lab procedure for potassium, again, we do the ISC method. It's the most common. The semipermeable membrane is selected for potassium. That membrane is valine-known-mycin. Again, the principle is the same as stated already for the chloride and sodium, and there are also direct and indirect methods for potassium. Your reference range is 3.5 to 5.1 millimoles per liter. The specimen type is serum or plasma or urine, and it can also be done in whole blood. The sources of error, of course, the most common one is hemolysis, and the release of potassium ions from the red cells will elevate your serum or plasma value. These samples that are this hemolyzed like this are not acceptable for the analysis of potassium. And that is the end of your presentation. Thank you.